Good day. My name is Lisa Wasko DeVetter. I'm an associate professor in the Department of Horticulture at Washington State University, and I'm joined by Emma Rogers, who is a research technician within my program and who did most of the work that you're going to be looking at today. The title of this presentation is Balancing Act: Impact of Pollinator Habitat on Pests in Blueberry Systems. And I'd like to acknowledge that we have several important contributors to this presentation. Dr. Brian Bernet, who is a aphid systemicist and contributed the information on some of the aphids that we're reporting on, as well as Donovan Bosnich, who's a master's student in Dr. Bernet's program at Carleton University. Also want to acknowledge the many collaborators that you see listed on this slide, or the institutions that they're affiliated with. They're involved in a much larger project addressing this question of how border habitats impacts pests in blueberry systems. And then I also want to acknowledge that this is a project funded by the USDA Multi-State Block Grant Program, and it is managed through Pollinator Partnership. So this is a presentation about pollination and pests. Let's first just go over why pollination is important and what pollination is. So pollination is the transfer of the pollen that is produced from the male part of the plants to the female receiving organ of the flower. It's really important in terms of being able to produce fruit and overall for food production. In fact, there's about 75 to 95% of flowering plants that are dependent on pollinators for pollination. And when we're talking about who's doing the bulk of pollination services, we're referring to insects primarily. Bees in particular are the ones that are doing most of the pollination services. So when we talk about pollination and managed systems like blueberry, a term that many of us are increasingly utilizing as we think about promoting and enhancing more resilient pollination systems is integrated crop pollination, or ICP. So ICP is a combination of various strategies, as you'll see on the figure on the right here. So we have horticultural practices, pesticide stewardship, and we also have utilization of managed honeybee hives. And this is very common for most blueberry production systems. Integrated crop pollination also can entail the use of alternative managed bees, such as managed bumblebee colonies or mason bees. And then what we're going to be focusing on a little bit more in the next couple of slides are the wild pollinators. So these are pollinating insects that could already be in your landscape or attracted to your landscape through integration of various practices, primarily through habitat enhancement. And that habitat enhancement is really important in terms of providing not just additional food sources beyond the blueberry crop that we're pollinating, but also nesting habitat as well. Now, wild pollinators are fantastic in many different production systems, and blueberry is no exception. There's many various species of pollinators that are wild that can contribute to blueberry pollination. Bumblebees are perhaps the most well-known, and bumblebees are important because they have the capability of buzz pollinating, or sonication, as some people refer to it. And what this is the vibration of flight muscles that triggers the effective release of pollen from the blueberry flowers. Honeybees are not able to do this, so there are some limitations in terms of the amount of pollen that a honeybee can extract and move from flower and flower relative to a bumblebee. So because of that, bumblebees are considered much more effective than honeybees on a single flower visiting basis. That being said, honeybees are still fantastic pollinators in blueberry systems and can get the job done because they have very populous colonies that we distribute in our fields. Again, bumblebees, they can be wild. They can also be managed. As you can see here, this is an image of a quad. BioBest and Coppert are just two companies that provide managed bumblebee colonies. However, there is some concern about utilizing these in systems such as blueberry, as well as other wild systems. They're great for pollination in greenhouses so long that they are enclosed and they don't escape. We have a little bit of concern about bumblebees escaping if they're in a managed colony situation and then incidentally and accidentally transferring viruses to and other types of diseases to wild populations, as well as possibly mating with wild populations and risking genetic diversity and contributing to genetic erosion. Another thing I want to point out on this slide too, so bumblebees nest in the ground. And so you can see here is a bumblebee queen in this diagram who's pollinating blueberry flower. She is nesting in the ground, and over time she will produce daughters and workers and mate, and then re-nest in the ground and overwinter. And then that new queen that is produced at the end of the season will then emerge in the spring, and the cycle will continue. But the important thing here that I want to point out is that this is an example of a ground nesting insect or a ground nesting bee. And that comes into play when I talk about conservation practices or conservation tillage. So how can you help pollinators? Well, it's actually really simple. Number one is just simply creating habitat around the field. And this image here from Pollinator Partnership does a really great job highlighting the various practices you can implement. So you'll see here where Mike's cursor is, there are habitat here on the border of this field. This can also provide a corridor to allow movement of bees and other types of beneficial insects around the perimeter of the field and across the landscape. And we really try to emphasize the use of natives when we're talking about additional habitat, because native plants tend to be more beneficial for native pollinators that could be present or attracted to your field. And some of these native species are even attracted to, some of these native species primarily benefit from these native plants, and native plants also tend to be a better resource in terms of pollen and nectar than some of the cultivated plant species. The other thing is to adopt integrated pest management, or IPM. Integrated pest management doesn't call for the complete ban of pesticides that are important at different times of the year, but it does encourage the utilization of more judicious planning and implementation of those pesticides. For example, scouting, looking at population thresholds, and deciding to make an application of a pesticide based on thresholds that are established for certain pest species. But we don't have those thresholds for all species of pests, which is part of the challenge that some growers have in terms of implementing an IPM program. The other practice to do is to engage in conservation practices such as conservation tillage, and that's important for those ground nesting bees that I mentioned previously, bumblebee being a fantastic example, and then providing clean water sources. There is some liquid that is provided by the nectar that bees forage upon, but they still really need more to be able to survive and reproduce. And so providing clean drinking water in the landscape is also critical. And then lastly, it's important to remember that although honeybees are important for providing pollination services and managed crops, including blueberry, having honeybees or honeybee keeping isn't necessarily equate to helping wild pollinators. In fact, there's some evidence that honeybees at really high densities can compete with wild pollinators for pollen and nectar resources in the landscape. So it's important to have a good density of honeybee colonies and not saturate your fields, because that might actually be detrimental to wild pollinators in your landscape. And then there are a few other examples here of good habitat, just to provide a few more visualizations. So up on the top left here, we're looking at, again, another hedgerow. Hedgerows tend to be primarily composed of woody species or perennial species, and are established in these linear strips around the perimeter of the fields, again, providing nice corridors for pollinators as well as other insects and organisms to move across the landscape. Then has an example here. This is an insectivorous strip, which tends to comprise of more herbaceous species as opposed to woody perennial species. But again, they can be established as linear strips around the perimeter of the field, or any open space that you might have that is available for planting habitat. And then there's also natural habitat, or sometimes referred to as semi-natural habitat. We have a lot of this in Whatcom County, and we've seen, based on other projects, that this semi-natural habitat can be a great refuge for wild pollinators that can contribute to blueberry pollination. Then there's programs that can help you learn about how you can establish habitat, and even provides certification to document that you're engaging in bee-friendly farming practices and providing habitat. Pollinator Partnership is one example where they have the bee-friendly farming program, and we'll highlight that again near the end of this presentation. But with that, I've covered some of the benefits of having habitat around a blueberry field for pollination services and some simple approaches to do that. Now we're going to talk about some of the downsides or the questions that a lot of growers have when it comes to establishing habitat and the barriers to adopting. And that's really the impact that these habitats might have on pest dynamics. So Emma, I'm going to hand it over to you and I'll let you present on this component of our presentation.
Awesome, thank you. So this question of what habitat enhancement means for pests inspired us to collaborate with Pollinator Partnership and run this project looking at this very question. There's not a lot of research surrounding this, so we teamed up with other universities and, like I said, Pollinator Partnership to um, figure out some answers to this question. So next slide, please. The first objective for this project was to monitor dominant pest communities in blueberry fields and in surrounding habitat margins. Here in Western Washington, we focused in on aphids because of their ability to vector virus, as well as spotted wing Drosophila because of their ability to lay their eggs in ripe fruit. So these are two of our most dominant pests in this region. So for this project, we focused in on those two pests. Throughout the talk too, I will be abbreviating spotted wing Drosophila as SWD or just spotted wing. So that's what that means if you see that later down the line. The second objective was to identify any beneficial insects that may be in the field or in the habitat margins. These are important because they will either predate or parasitize some of these pests and are a great thing to be seeing in your fields. And lastly, we want to leverage these findings to create a free and available habitat planting recommendation tool that you can access. And if you're interested in planting pollinator habitat, to be able to see what that means for associated pest species. And we will highlight that tool a little bit later in this presentation. Next slide, please. As I mentioned, we've collaborated with a few different universities and Pollinator Partnership. So here in Western Washington, we are carrying out the project with Washington State University. Oregon State University was also involved, as well as Pollinator Partnership in California, University of Michigan, and the University of Vermont as well. Next, please. So here on the West Coast, all three of our states were focused in on aphids and SWD for those reasons I mentioned earlier. Next, please. In Michigan, they were focusing in on cranberry fruitworm, blueberry maggot, and SWD. Next, please. And then Vermont was looking at Japanese beetle, blueberry maggot, and SWD. So SWD is a prevalent pest across the entire country. So this was some really important research to be carried out here. Next. For our site and study design here in Washington, we had 5 sites, 2 of which were organic and 3 were conventional. Within a field site, we had 4 in-field sampling locations, 2 along the edge and 2 in the interior of the field. We also had 6 habitat or border plant sampling locations. We tried to follow a similar scheme where we had some along the edge and some in the interior of the habitat. So we attempted to have an edge and interior pair where possible. I think if you flip to the next animation, we'll highlight that. We tried to have that pair where possible, but sometimes there were, water barriers or the vegetation was too thick and we were not able to make it into the interior of the habitat, which in that case, we just put our sampling locations along the edge, as you kind of see on the left there. This data was collected in 2023 and in 2024, which completes the 2 years of data collection for this project. Continuing on, I will only be focusing on the data for 2023. We don't quite have the 2024 data processed yet, but that will be coming in the future. Next slide, please. We had 2 different types of methods, sampling methods for this project, both passive and active. So for the passive methods, we have the yellow sticky cards, and this was to target any aphids. So these were hung up in the field and left out for 2 to 3 weeks at a time when we came back and changed them out. We also have, if you go to the next, is the red sticky cards with a pheromone lure to attract Drosophila species. So these red sticky cards were aiming to target SWD. And again, hung up in the field, left out for 2 to 3 weeks. So both of these types of cards are meant to see what is passively flying through the field, crawling on these plants. We couldn't make a species-specific determination, especially in the habitat plants, but we wanted to get a general sense of what was going on in the field. And then we also had some active sampling methods as well. So for aphids, we did a leaf turn sampling method where we sampled randomly 20 leaves at each sampling location, flipped them over, and counted any aphids that we found. So if we found live aphids, they were counted and also collected and shipped off to Brian Brunet in Canada for species identification. So I'll present some data on that in a little bit. As far as actively sampling for SWD, we collected any ripe fruit we found in the field whenever it was available. So we collected blueberry, as well as from some of our habitat plants and salmonberry, blackberry, huckleberry, honeysuckle, etc. And we took them back to the lab and did a larval salt extraction. So we were able to see and count if there were any SWD larvae living in the fruit when we collected it. And then the last active sampling method we have was sweep net sampling that we did on all of our habitat plants, the dominant species. And this was to give us an idea of what is on a specific species at the point of sampling. So this way we could determine if there were any beneficials living on these plants or any pests, etc. Next slide, please. So to jump into some data, I want to remind you again, this is only for 2023. These graphs you're looking at now are also an average of all sites and all sampling locations broken down between conventional and organic management. On the left, you have the average aphid count per card on the y-axis with the month going along the x-axis. So the graph on the left is for the yellow sticky cards. A reminder also that both of these graphs come from our passive sampling methods. So the graph on the left is the aphid counts, and then the graph on the right is the SWD counts. What we can see from this, the SWD have a pretty linear trend increasing throughout the season, whereas the aphid dynamics and the trends we're seeing across the season with the aphids is a little different here. So I wanted to kind of untangle that a little bit more. If we go to the next slide. I guess we're going back to SWD first, and we'll get into aphids in just a moment. But I did want to highlight that pest dynamics are site dependent. When we break down these red sticky cards between each of our conventional sites, so you can see one site's trends on its own graph, we see the similar trend that we saw broken down across the management style here. I wanted to note too, this legend, we go from crop interior to crop edge to habitat interior, trying to capture this gradient from what's happening inside the field to the edge to the inside of the habitat. And if we go to the next slide, this is where it's really important to look at these data on a site-by-site basis. These data are for the yellow sticky cards, again, with average aphid count per card on the y-axis, but now broken down site by site for each of our conventional sites. And you'll see in the conventional 1 site, we're seeing more aphids on the crop edge, but if we look at the conventional 3 site, we're seeing more along the habitat edge. And if you go to the next animation, I just want to highlight what is happening here in this conventional 3 site. We're finding some aphids in the field, as well as a higher instance of them on the habitat edge. But if we go to the next slide, we're going to compare our active sampling method, the yellow sticky card, to the passive sampling method on the left, the yellow sticky card, to our active sampling method, the leaf turns on the right graph. So the left is the same with average aphid count per card on the y-axis, and then on the right you have average aphids per 20 leaf sample. And this again is for 1 field site. So on the left we're seeing those aphids in the field on those yellow sticky cards, but then when we were actually scouting for aphids at the time of sampling, we did not find any on any of the blueberry plants whatsoever. Those on the habitat were still being captured, but not on the blueberry plants. And the takeaway from this is that there may be aphids seen in the field, but they may not be using blueberry as a host, and so therefore not vectoring any virus in your field. Next slide, please. So then we can look at this similarly with the SWD, where on the left we have our passive sampling method, the red sticky card, where you are seeing some SWD in the blueberry crop. But then when we extracted larvae from the fruit in our active sampling method, we weren't finding any in blueberry whatsoever. We only found some in the habitat edge, and if you go next, this will tell you what plant it was, and it was on blackberry. SWD notoriously love blackberry, but the good thing to note here is that conventional management is doing a good job at controlling for this SWD in the blueberry fruit specifically. Next slide, please. So this is our data that we received back from Brian Brunet in Canada. And so he went through and did a molecular DNA analysis to break down these aphids that we sent him to a species level. So on the left, you can see in blueberry, he found two aphid species, Ericaphis fimbriata and Illinois azalea. Blueberry is a known host for both of these, so there was nothing too shocking there. And then if we look on the right at the border plants, you'll see the different border plants that these aphids were collected from, and then the aphid species that were found on those plants. And you'll notice that there are the specific species for that plant that use that plant as a host, and there's not a lot of spillover between blueberry and the border plants. There is one I wanted to note, which is the Ericaphis fimbriata that was found on rose, but rose notoriously has a lot of species that are polyphagous, which means they have multiple hosts. So this is likely a spillover from blueberry onto the rose versus the other way around. And again, all of these species that we found were associated with the known host plants as expected, so nothing too shocking there, which is nice. I think the biggest takeaway from this data is that there was no spillover from the border plants, any of those species there, into blueberry. If we go to the next slide, some of our conclusions from this data. Organic fields have higher instances of SWD, especially compared to conventional fields. Pest dynamics are site dependent, particularly regarding aphid abundance. The SWD had a little bit more of a similar trend across sites, but looking at the aphid abundance, it's to tell the full story, you need to look at this on a site-by-site basis. This led us to see that even though we were finding aphids in the field, they were probably not using blueberry and therefore not vectoring virus, because when we did our active sampling and scouting, we were not finding any aphids on blueberry in the crop in these conventional fields. Similarly, conventional management is doing a good job at controlling SWD in the fruit as well, because we found no SWD larvae in any of the blueberry that we collected from our conventional fields. And again, the biggest conclusion I think that we found is that there was no aphid spillover from these habitat plants into blueberry. So these are plants that are great for pollinators. They offer a lot of forage and habitat, like Lisa had highlighted earlier. And in the cases of our fields, there was no aphid spillover, at least, into the crop. Next slide, and we'll pass it back to Lisa, and she will highlight the tool that this data is going to be used for.
Great. Thanks, Scott. Thanks, Emma. Really a lot of great information, and more information will come over time. But again, hopefully the information that we presented will give land managers more confidence to be able to implement habitat and be aware that habitat will not be increasing their risk of aphids that could vector viruses like scorch, as well as spotted ring Drosophila, as long as they stick with their recommended management practices. So if you're looking at establishing habitat or want to learn more about what types of pests might be associated with habitat already present in your field, there's a really great tool that came out of this project. This is the Pollinator Enhancement Security Tool, or the PEST tool. This is available for free for you to utilize. If you scan the QR code or visit the website that you'll see listed here, this will take you directly to this particular tool. There's various facets of the tool, but what we wanted to highlight today was two components. So first, we'll look at, what are the particular plants that might be in your habitat or adjacent to your field, and what the level of risk might be for key pest species. So you'll see here, we've picked salmonberry as an example, very common here in the borders of some of our fields within the Pacific Northwest. And you'll see that spotted wing Drosophila is known to be a pest that will utilize salmonberry. You can also look at it in the opposite way. You can focus on it with attention to a specific pest that you might be concerned about. If you click on this icon and enter the pest that you're concerned about, in this example, we chose spotted wing Drosophila, and if you select that, what will be populated is the known hosts for spotted wing Drosophila, as well as some of the beneficials that might predate or parasitize spotted wing Drosophila. So really good tool. It's going to be augmented and enhanced too with the data coming from this particular project in Washington and other states. So it's not a Washington specific tool. It's a tool that I think will be useful anywhere that blueberry is being grown in North America or in similar latitudes with similar pest dynamics. There's additional resources for you to consult with if you're looking to establish habitat. Certainly the conservation districts in Whatcom County, Skagit County, and elsewhere within Washington State or other states are a great place for you to stop at and learn more about if there's easement programs or other types of programs that can help you economically establish some of the habitat. There's also extension offices such as Washington State University Extension is happy to provide support. And then I mentioned there's programs such as the Bee Friendly Farming Program through Pollinator Partnership. And then the Xerxes Society has a similar type of program. I'm the current manager of the Bee Friendly Farming Certificate at Washington State University at the Northwestern Washington Research and Extension Center. And I've managed it for 2 years now. It's very cheap. It's a $45 annual fee, and it's also pretty easy to implement. So you don't have to change a lot of your practices. The key thing that this certification program will be looking for is provision of habitat, as well as provision of water, as well as some other resources. So things that we were already doing at the station, and I would surmise a lot of other growers are already doing or could simply implement to show that they're then engaging in bee-friendly farming practices. So something to check out if you're interested in. And then last but not least, projects like this are large and rely on a lot of collaborators from various disciplines. We're a horticulture lab, both Emma and myself, and so we have a lot of gratitude to extend to the entomologists that work with us on this project. But we had other players within the Small Fruit Horticulture Lab that contributed. So we had Caleb Brower, Selena Helmer, Aidan Williams, Heather Pedroza, and Brian Malpin, as well as Micah Evault. They are all students and technicians and time slip workers that contributed to data collection. We also want to thank Jana Lee, who is at the USDA ERA Center in Corvallis, who trained Emma on the identification of some of the pests as well as some of the beneficials. And hopefully we'll hear about some of that results of the beneficial work in future presentations. Louis Armstrong, who runs the Horticultural Crop Research Entomology Program at the WC Mount Vernon Research and Extension Center, was also critical, as well as Ben Deal for providing some on-the-fly entomological and taxonomic assistance. And then, of course, Pollinator Partnership, collaborating universities, and the growers that allowed us access to their fields over the past 2 years to collect data to answer some of the questions spurred by this research. With that, I want to thank you all for your attention. If you have any questions, don't hesitate to reach out. There's ways to reach out. You can just search on WSU Small Fruit Horticulture Program, and you should be able to find my contact information there since it's not listed on this slide. With that, thank you and goodbye.